Hello, good evening everyone. Today we will be practicing questions on the topic forest genetics, tree breeding, seed technology, biotechnological applications. Let us start without any delay. Question number 1. The main objective of forest genetics is option A, increased tree height, B, improved productivity and adaptability, C, reduced forest fires, and D, improved soil fertility. So correct answer is the main objective of the forest genetic is to improve productivity and adaptability. Question number 2. The branch of forestry that deals with inheritance of traits in trees is called option A, silviculture, dendrology, forest genetics, and agroforestry. So the branch of forestry that deals with inheritance of traits is forest genetics. Option C is correct. Question number 3. Plus trees are selected based on option A, poor growth, superior phenotype, random selection, and soil fertility. So it is selected based on superior phenotype. Question number 4. Which is commonly used vegetative propagation method in forest tree improvement? In forest tree improvement, which of the following is used? So yes, grafting is used is correct. Layering is also used. Tissue culture is also used. So all of the above is correct. Question number 5. Provenance trials are conducted to test option A, fertilizer requirement, soil erosion rate, genetic variations among populations, and option D, fire resistance. Question number 5. So provenance trials are conducted to test genetic variations among populations. So option C is correct. Question number 6. Hybridization in forest trees is mainly used to. Hybridization in forestry is mainly improved to option A, increase photosynthesis, combine desirable traits, reduce pollination, and improve soil. So it is mainly used to combine desirable traits is the correct answer. Don't worry, I will give the answer key at the last. Before that, जिन्होंने भी सब्सक्राइब नहीं किया वो सब्सक्राइब कर सकते हैं for further updated videos. Question number 7. Controlled pollination is essential in question number 7. Natural regeneration, tree breeding, seed dispersal, and copy sizing. 7 is controlled pollination is essential in tree breeding is the correct answer. Question number 8. The first step in tree breeding program is which of the following is the first step? Hybridization? No, this is not the first step. Selection of plus trees, yes. Selection of plus tree is the first step for breeding program. Most important question. Question number 9. Which of the following is a method of clonal forestry? Clonal forestry. Seed orchid, wrong. For clonal forestry, micropropagation is the correct answer. Question number 10. A seed orchid is established primarily for recreation, research only, production of genetic improved seed, and timber. So correct answer is production of genetically improved seed is the correct answer. Question number 11. Seed viability refers to the ability of seed to germinate, color, seed coat, seed size, and seed dispersal. So correct answer is the ability of the seed to germinate is the correct answer. Question number 12. Orthodox seed cannot be stored for. Orthodox seed cannot be stored for. Orthodox seed can be stored for, no? So this is not correct. Short term only, yes. It can be stored for long term under low temperature and humidity, yes. For orthodox seed, we need low temperature. So option B is correct. Question number 13. Recalcitrant seeds are: option A, drought resistance, cannot tolerate desiccation in storage, easy to store and genetically modified. So recalcitrant seeds cannot tolerate desiccation and storage. So B is the correct answer. Question number 14. The standard temperature of long-term seed storage in gene banks is 5 degrees Celsius, 0 degrees Celsius, and 20 degrees Celsius and 20 degrees Celsius. So for long-term seed storage, we need very low temperature, which is minus. So minus 20 degrees Celsius, which is lower temperature. So question number 15. ISTS stand for what? International Seed Testing Association. International Seed not technology, it is International Seed Testing Association. Option B is the correct. Question number 16. Somatic embryogenesis is important in forestry for: option A, hybrid seed production, clonal propagation through tissue, natural regeneration, and pollen storage. 16 is clonal propagation through tissue culture is the correct answer. Molecular makers are used for: option A, forest fire control, genetic diversity studies, seed drying, and seed fertility test. The correct answer is genetic diversity studies. Cryopreservation is a method of seed dispersal, long-term conservation of genetic material at low temperatures. Cryopreservation means at very low temperature. So option B is the correct. Question number 19. The main advantage of tissue culture in tea improvement is: option A, produce identical clones rapidly, B, increase seed size, reduce soil erosion, and enhance timber density. So correct answer is, the main advantage is produce identical clones rapidly is the correct answer. Question number 20. Genetic engineering in forestry can help to develop fireproof trees. Yes, it is also true. Pest resistant trees, this is also true. And drought tolerant varieties, this is also true. So all of the above is correct. Question number 21. The science of improving the genetic quality of trees is called: what is called? Is called tree improvement is the correct answer. Question number 22. The technique of crossing individuals with different genetic backgrounds is: with different genetic backgrounds. The technique of crossing: inbreeding, outbreeding, hybridization, and mutation. So correct answer is hybridization. Which method ensures uniformity in forest plantation? Seed propagation, vegetative propagation, natural regeneration, and grazing. So 23. It's the best. Which method ensures uniformity is vegetative propagation. Question number 24. Clonal seed orchid is developed using option A, seed of plastris. False. Grafted clones of plastris is the correct answer. Grafted clones of plastris is the correct answer. Question number 25. Progeny test is conducted to evaluate 25. Progeny test is conducted to evaluate growth rate of trees. Incorrect. Genetic worth of parent trees is the correct answer. Question number 26. Which one is not a method of vegetative propagation? Grafting correct, layering is also correct, tissue culture is also correct, but broadcasting is not the method. So this is correct. Question number 27. Which of the following is a tree breeding objective? Tree breeding objective. Pest resistance, high wood quality, fast growth. So all of the above is correct. Isogymes are used in soil fertility testing. No. Isogymes are used in genetic diversity analysis. Is the correct answer. The National Bureau of Plant Genetic Resource (NBPGR) is located in New Delhi. Is the correct answer. The gene bank in India for forestry species is mainly managed by ICFRE. Is the correct answer. These are direct questions of NCERT, so I'm not discussing much about this. The seed that cannot withstand drying and freezing is called recalcitrant. The moisture content of orthodox seeds for long storage is 5 to 7%. Is the correct answer. The viability test during color change in seeds is float test, tetrazolium test, germination test, and X-ray test. So the viability test will be tetrazolium test. Is the correct answer. Seed dormancy is mainly, question 34. Seed dormancy is mainly caused by hardcore seed. Is the yes, this is correct. Seed immature symptoms is correct. Immature embryo is also correct. Chemical inhibitors is also correct, and also all of the above. Question number 35. Pre-sowing treatment in given to seeds to increase question number. Pre-sowing treatment is given to seeds to question number 35 to increase dormancy. No. Pre-sowing treatment is given to the seeds to break dormancy and improve germination. Is the correct answer. So this was our last question of this session. If you want more videos like, then please comment and subscribe to the channel.